Welcome to the Deep Dive, where we sift through the sources, find the essential knowledge, and deliver the profound insights straight to you.
Today, we are tackling what you could argue is the very definition of life itself, self-reproduction. We're going to be exploring the incredible, highly conserved choreography that governs cell division in complex organisms, the eukaryotic cell cycle.
Okay, so let's unpack this right away. The core question is just staggering.
Yeah.
How does a single fertilized egg coordinate everything inside it to grow, perfectly copy its entire genetic library, which is, what, kilometers long if you unspooled it?
It's massive.
And then it has to cleanly distribute those huge packages of chromosomes to produce two perfect daughter cells. The numbers alone. I mean, the sources point out that from one single cell, you get an adult human built from approximately 10 to the power of 14 cells.
And trillion cells.
That degree of controlled, accurate division requires an almost absurd level of molecular precision.
It's absolute military precision. And our mission in this deep dive is to shortcut you directly to the molecular machines that enforce that precision. We want to understand not just the timeline, but how the cell makes sure that step A is totally finished before step B even thinks about starting.
Right.
The entire regulatory system is conserved across all eukaryotes, and it all hinges on this sophisticated, integrated network of molecular switches, specifically a type of enzyme called protein kinases.
And this isn't just abstract biology. This is where it gets incredibly important for all of us. Defects in this exact conserved control machinery, these protein kinases and their partners, are common and really a fundamental cause of cancer.
Absolutely. When the cell loses control over its division, proliferation just runs rampant.
So understanding the highly regulated proper function of the cell cycle it's the baseline for diagnosing, treating, and really understanding disease.
It is. So we're going to trace the path from the outside commands, things like environmental signals and growth factors in higher eukaryotes, down to the core intrinsic machinery that governs progression through these vital tech points. Let's start at the very beginning, the timeline.
The timeline. So when most people think about a cell dividing, they probably picture that final dramatic stage, right? Mitosis.
The chromosomes pulling apart.
Exactly. But that's just a brief finale. The entire eukaryotic division cycle is this coordinated sequence of four big processes: cell growth, DNA replication, chromosome distribution, and then finally division itself.
And that coordination is what really separates eukaryotes from simpler organisms like bacteria. In bacteria, growth and DNA replication are often just ongoing. They're linked in a continuous fashion.
But not in our cells.
No. Eukaryotes separate the process into discrete, strictly timed events. We basically break the cycle down into two periods: the highly visible M phase, which is mitosis and cytokinesis,
The dramatic part.
and the much, much longer period known as interphase.
And M phase is remarkably brief. For a typical human cell, we're talking about only an hour. So if a cell cycle is 24 hours long, that means, what, 95% of the cell's life is spent in interphase?
That's right, 95%. And during interphase, if you look at a nucleus under a standard microscope, it appears pretty uniform morphologically. And that's because the chromosomes are decondensed. They're all spread out.
Which is essential, right? They have to be loose and accessible for the cell to read them and, more importantly, to copy them.
Exactly. That decondensed state is what allows that massive amount of DNA to be accessed and accurately replicated during the next crucial stages.
And those stages divide interphase into three distinct intervals. They're all named around the synthesis, or S, of DNA. And while the cell is growing steadily through all three, G1, S, and G2, the specific biochemical jobs are completely different.
So we begin with G1, or gap 1. This is the interval right after the previous division, M phase, and before the start of DNA replication. During G1, the cell is intensely metabolically active. It's synthesizing proteins, it's making new organelles, it's growing. But it is not yet copying its genome.
Then comes S phase,
Yeah.
the synthesis phase. This is the period dedicated 100% to DNA replication. The cell has to accurately duplicate every single one of its chromosomes, and this has to be completed perfectly before it can move on.
It has to be perfect. Following that, we have G2, or gap 2. This is the interval between the end of S phase and the start of M phase. The primary focus here is preparation for the big show.
So last-minute checks.
Exactly. It's synthesizing specific structural proteins, tubulins for the spindle, the mitotic kinases, everything required to build the complex machinery of mitosis.
And if we look at typical timing, let's say a rapidly dividing human cell in a lab culture with a 24-hour cycle. G1 is the longest, right? Maybe around 11 hours.
Yeah. That's all.
S phase is pretty substantial. It takes about 8 hours. G2 is shorter, maybe 4 hours. And M phase, the grand finale, just takes that final hour.
Right, but these timings are incredibly flexible, and they tell us a lot about what the cell is prioritizing. For example, budding yeast. They're focused purely on speed. They can complete all 4 stages in just 90 minutes.
Wow, 90 minutes. So they must have very short G1 and G2 phases.
Extremely short. And the sources give an even more striking example. Early embryonic cell cycles, right after fertilization. They can whip through a full cycle in 30 minutes or even less.
How is that even possible?
They basically skip G1 and G2 entirely. They just alternate between S phase for rapid DNA replication and M phase for division.
Ah, because the original egg cell is massive. It's already packed with all the proteins and components it needs.
It's preloaded. The goal here isn't growth or gathering resources. It's the rapid partitioning of that huge cytoplasm into lots and lots of smaller cells. These accelerated cycles really show you that G1 and G2 are, well, they're regulatory periods. They aren't strictly essential for the mechanical act of division itself.
Okay, but this all assumes the cell is dividing. What about the vast majority of cells in an adult? Most of them seem to have exited this process completely.
And that defines the quiescent state, or G0. A cell can exit from G1 to enter G0. Now, while it's in G0, the cell is still metabolically active. It's still doing its job, whatever that might be.
But it's not preparing to divide.
Exactly. It ceases growth, it significantly reduces its rate of protein synthesis, and it just stops proliferating. It hits the pause button, potentially indefinitely.
And what happens to a G0 cell can vary a lot. Some cells, they permanently withdraw. Think of most of your adult nerve cells or muscle cells. They're never going to divide again, barring some kind of catastrophic failure.
Right, but many others, like your liver cells or skin fibroblasts, they're just resting. They can be triggered to re-enter the cell cycle, to go back into G1 if they get the right stimulus.
Like the growth factors released during an injury to promote wound healing.
That's a perfect example.
So, to study this whole dynamic system, researchers need a way to accurately figure out which stage a cell is in at any given moment. This brings us to a really powerful technique, flow cytometry.
Flow cytometry it's beautiful in its simplicity for this kind of analysis because it relies entirely on tracking one thing, the quantity of DNA inside the cell. It's just a quantitative measurement.
Okay, so walk me through it. If we start with a standard diploid cell in G1, it has 2 copies of each chromosome. We can quantify that as $200 DNA content.
Exactly, $200. Now, once that cell goes through and completes S phase, it has duplicated its entire genome. But those 2 complete genomes are still housed within a single nucleus.
Ah, so cells in G2 and M phase have precisely double the DNA. They have a $4 content.
Precisely. And the cells that are in the middle of S phase would have a DNA content somewhere on a gradient, continuously increasing between $200 and $4. The technique uses a fluorescent dye that binds to the DNA, so the machine can just measure the fluorescence intensity of thousands of individual cells per second. And when you look at the data plot from this, the sources describe it really vividly. You see 2 distinct sharp peaks.
Okay, so the first, usually larger peak, that's the G1 cells with $200 DNA content.
That's them. The second, smaller peak corresponds to the G2 and M cells, all with 4R long DNA content.
And the S phase cells, they must be the smear or the distribution of fluorescence intensities that connects those 2 peaks.
Exactly right. This method lets scientists not only calculate the proportion of cells in each phase, but through sorting, they can actually isolate cells that are currently at a specific point in the cycle for more in-depth molecular study. It's quantifiable proof that these phases are truly discrete, coordinated events.
Okay, so that coordination is staggering. To go smoothly between G1, S, G2, and M without making a single mistake, it requires the cell to listen to 2 main kinds of signals, external cues like, is the environment right?
Or is my neighbor telling me to divide?
And internal monitoring. Things like, is my DNA finished replicating and is it intact?
These extrinsic and intrinsic signals all funnel down and converge on these critical regulatory points that are designed to prevent errors. The sources really emphasize 2 main commitment points in the G1 phase, one for simple eukaryotes and one for animals.
Let's start with the one in budding yeast, Saccharomyces cerevisiae. It's known as START.
START it's the point of no return. Once a yeast cell passes START, it is metabolically and genetically committed to completing the entire division cycle, even if the external conditions suddenly get worse.
And in yeast, what controls that decision?
It's controlled by 2 very pragmatic factors. First, is there enough food-adequate nutrient availability? And second, has the cell reached a critical size? If food is scarce, the cycle arrests right there in G1 before start.
And the cell size thing is a really elegant feedback mechanism, isn't it? Especially because budding yeast division is unequal. You get a big mother cell and a smaller daughter cell.
Exactly. To ensure the health of the population across generations, that little daughter cell has to spend a significantly longer time in G1, growing, before it's allowed to pass start and start copying its DNA. Start acts as an internal timer based on cell volume.
Okay, so that's yeast. For animal cells, the analogous G1 control point is called the restriction point.
Right. And this mechanism responds less to simple nutrients and much more critically to external signals, specifically extracellular growth factors.
That's the key difference.
It's the critical functional distinction. If the right growth factors are present and they're telling the cell to divide, the cell will pass the restriction point and commit to S phase. And that commitment is total. The key insight is that once it's past this point, the cell is entirely self-sufficient. It will finish the cycle and divide even if you take those external growth factors away.
So if you remove the growth factors before it passes the restriction point,
Then it immediately halts progression and enters that quiescent state, G0, to wait for a new signal. The clinical example is perfect. Platelet-derived growth factor, PDGF, is released when blood clots. That signals nearby fibroblasts to quickly exit G0, pass the restriction point, and divide to make new tissue for wound repair. It's a system that's on demand.
now while G1 is the main decision point for most of us, the sources mention that some systems actually place their primary control at the G2 to M transition.
Yes, this is common in fish and yeast, Schizosaccharomyces pombe, where size and nutrients are monitored right before mitosis. But the most striking animal example involves vertebrate oocytes, are egg cells.
Right. They can remain arrested in G2 for decades.
Decades in humans, just waiting, until a hormonal signal finally triggers the cell to proceed straight into M phase to complete meiosis. It just shows the system has evolved multiple effective arrest points.
So the external signals are like the green light, but the cell needs its own internal guardrails, the cell cycle checkpoints, to make sure the internal machinery is actually working.
The primary job of these checkpoints is quality control. It's all about enforcing ordered progression and preventing catastrophic outcomes. The absolute cardinal sin for a cell is to start the most violent process, mitosis, before the most sensitive process, DNA replication, is 100% complete and repaired.
So we have the DNA damage checkpoints. These are sophisticated monitoring systems that are active in G1, in S, and in G2. They detect damage or incomplete replication, and they coordinate a cell cycle arrest with simultaneous DNA repair.
The G2 checkpoint, for instance, it prevents the cell from even starting M phase if there's still damaged DNA or if the genome hasn't fully finished replicating.
And the G1 checkpoint is just as vital. Arresting there allows for repair before you enter S phase. That prevents a damaged template from being copied and locking that mistake into the genome forever.
And then the S phase checkpoint provides continuous active monitoring during the replication process itself. If there are errors or the replication fork stall, the whole process pauses, repair machinery comes in, and integrity is maintained before synthesis continues.
Okay, then at the very, very end of the cycle, right near the end of mitosis, there's one last specialized check for the physical distribution of the chromosomes.
That is the spindle assembly checkpoint, or the SAC. This is absolutely essential for genome stability. The SAC operates during metaphase, and it monitors whether every single kinetochore, the attachment point on each chromosome, is correctly attached to microtubules coming from opposite poles.
So if even one chromosome is dangling or attached incorrectly.
The checkpoint issues a powerful arrest signal. It locks the cell in metaphase. It's the ultimate all-clear signal. It prevents the physical separation of the sister chromatids, the key event of anaphase, until accurate segregation is absolutely guaranteed. It ensures each daughter cell gets a perfect, identical set of chromosomes.
This coordinated control is just stunningly precise. But for decades, the question was, what is the molecular engine driving all this? What's the chemical signal?
And our current understanding, amazingly, comes from 3 initially separate but ultimately convergent experimental paths that use completely different organisms.
It's a classic story of conserved biology. Research on frogs, yeast, and sea urchins all pointed to the exact same core mechanism, a system that's been functional for maybe a billion years.
Let's start with the frog oocytes. That's where the original biological activity was discovered. Frog oocytes naturally arrest in G2, and their entry into M phase is triggered by the hormone progesterone.
Okay.
So in a landmark 1971 experiment, researchers took cytoplasm from a hormonally stimulated oocyte, one that had already entered M phase, and they microinjected it into an unstimulated G2-arrested oocyte.
And the recipient cell?
Yeah.
It just jumps straight into M phase.
Immediately. It completely skipped the hormone requirement. The conclusion was that some diffusible factor in the cytoplasm was entirely sufficient to trigger the G2 to M transition. They named this activity maturation-promoting factor, or MPF, and it was later shown to be the general regulator of M phase entry in all eukaryotes.
Amazing. Okay, path 2 takes us to yeast genetics, pioneered by Leland Hartwell and Paul Nurse in the 1970s.
Right. They used temperature-sensitive mutants, the CDC mutants, cell division cycle mutants. These would grow normally at one temperature, but if you shifted them to a higher, non-permissive temperature, their cell cycle would arrest at a very specific point.
Which allowed them to isolate the exact genes responsible for controlling those specific transitions.
Exactly. The key discoveries were the genes CDC28 in budding yeast and CDC2 in fission yeast. CDC28 was required for the cell to pass start, that G1 commitment point, and the homologous gene, CDC2, was required for both the G1 transition and the G2 to M transition.
And when they cloned these genes, what did they find?
The critical discovery. They encoded a protein kinase. This was the first concrete evidence that cell cycle progression was governed by phosphorylation, the enzymatic addition of phosphate groups. The conserved protein kinase from these genes is now known as CDK1.
Okay, so we have the MPF activity from frogs, the CDK1 kinase from yeast, and the third piece of the puzzle came from sea urchin embryos.
Right, which are a fantastic model because they divide synchronously and really rapidly after fertilization. And researchers knew these divisions required new protein synthesis.
So they went looking for those proteins.
In 1983, Tim Hunt and his colleagues identified two proteins, cyclin A and cyclin B, that showed this dramatic periodic fluctuation. He named them cyclins because they accumulated steadily all through interphase, but then were instantly and rapidly destroyed near the end of mitosis. A cycle. Hunt immediately suggested this oscillation, this appearance and destruction, was what controlled the entry and exit from M phase.
So you've got the MPF biological activity, the CDK1 protein kinase, and the oscillating cyclin protein. The convergence was just inevitable.
It was. In 1988, James Mahler's lab purified MPF from frog eggs and found it wasn't a single protein. It was a dimer.
Two parts.
It was made of CDK1 and cyclin B. The cyclin B acts as the necessary regulatory subunit. CDK1 by itself is inherently inactive. It only gains its kinase activity when cyclin B binds to it. No cyclin, no activity.
That's brilliant. But it raises a new question. If CDK1 and cyclin B start forming complexes in G2, how does the cell prevent M phase from starting too early? You need some kind of activation switch.
You do, and that's where antagonistic phosphorylation comes in. It's absolutely essential for creating that sharp, sudden transition into mitosis. When the CDK1 cyclin B complex forms in G2, it is immediately phosphorylated on two different sites.
Okay.
One phosphorylation on a threonine residue is actually activating, but two other phosphorylations on a tyrosine and another threonine are inhibitory.
So one is a go signal and the other is a stop signal, creating a kind of locked, waiting state.
It's perfectly put. The stop signal phosphorylation is done by a protein kinase called WEE1. So the CDK1 is there, it's complexed with cyclin B, but it's sitting there, doubly phosphorylated and locked in this inactive state.
So to fire the starter pistol and push the cell into M phase, you have to remove that inhibitory brake that WEE1 put on.
And that job belongs to a family of enzymes called the CDC25 phosphatases. When the cell is finally ready, it's the right size, the DNA is intact, CDC25 swoops in and removes those inhibitory phosphates. That sudden dephosphorylation is what dramatically activates the MPF complex, driving the cell sharply over the G2M threshold.
Okay, so now mitosis is underway. The cell has to ensure a rapid and irreversible exit to get back to interphase.
Yeah.
The CDK1 cyclin B activity has to be shut down just as quickly as it was turned on.
And this involves a beautiful self-regulating feedback loop. The active CDK1 cyclin B complex doesn't just drive M phase events, it also triggers its own destruction.
How?
It does this by activating a powerful ubiquitin homologous complex called the anaphase-promoting complex cyclosome, or APCC. You can think of the APCC as the cell's molecular grim reaper.
And the APCC gets an execution order for cyclin B.
Yes. The APCC tags cyclin B for destruction. It's hauled off and instantly degraded by the proteasome. The moment that regulatory cyclin B subunit is destroyed, CDK1 instantly loses its catalytic activity.
And that sudden, irreversible inactivation is what lets the cell exit mitosis.
Exactly. It allows the cell to reform nuclear envelopes, decondense its chromatin, and enter the next interphase. It's a commitment to self-destruction to reset the entire system for the next round.
That initial CDK1 cyclin B complex is the prototype. But in complex animals like us, the system is much more diversified. We now know that CDKs are cyclin-dependent kinases, and their activity is tuned by specific cyclin partners for different jobs.
In simple yeast, CDK1 is the only CDK. It just associates with different G1 cyclins to pass start, and then B-type cyclins for S and M phases. It's like a single engine with different keys.
But animal cells use a whole fleet of engines, multiple CDKs and cyclins to precisely coordinate every single transition point. It allows for much finer control based on all those external signals.
We have a pretty clear functional map now. In early G1, the cell relies on CDK4 and CDK6, complex with cyclin D. These are the crucial kinases that respond to growth factors and drive the cell through the restriction point.
Then, for the critical transition from G1 into S phase, the actual initiation of DNA synthesis, the cell activates CDK2 partnered with cyclin E.
As the cell progresses through the hard work of S phase and G2, CDK2 switches partners to cyclin A. And finally, the G2 to M transition and the major events of mitosis are driven by that original MPF complex. CDK1 complexed with cyclin A and cyclin B. Each specific CDK-cyclin pair phosphorylates a different specialized set of target proteins needed for that phase.
So cyclins are the required activators, they're the fuel. But if the cell needs to hit the brakes hard, it uses specialized inhibitors.
That's the third essential layer of regulation, the CDK inhibitors, or CKIs. And mammalian cells use two major families of these inhibitory proteins.
Okay, what are they?
First, we have the INK4 family proteins like P15 or P16. They're highly specific. They only inhibit CDK4 and CDK6. By targeting those G1 CDKs, they block progression through the restriction point.
And the second family?
The CYPKIP family. This includes P21, P27, and P57. These are much broader inhibitors. They primarily block the activity of the CDK2 cyclin E or A complexes. By controlling the synthesis and degradation of these CKIs, the cell has this precise, rapid control over whether the cycle moves forward or just stops in its tracks.
Let's really focus now on how those outside signals, the growth factors, actually lead to the cell's internal commitment to divide. How do they drive it past the restriction point and into S phase? This is where external permission translates into internal action.
The central mechanism is, well, it's beautiful in its simplicity. Extracellular growth factor stimulation, often relayed through pathways you might have heard of like Ros-Raf-Meckerk, rapidly induces the synthesis of cyclin D.
And cyclin D is unstable, right? It's rapidly degraded by the APCC, so you can only maintain high levels of it as long as the cell is receiving continuous external growth factor signals.
It's an immediate, real-time indicator of whether the environment is favorable for division. If those growth factors are removed before the restriction point is passed, cyclin D levels plummet, CDK46 activity drops, and the cell can't proceed. It's forced into G0.
And that tight link is why dysregulation of cyclin D, either making too much of it or inactivating its specific inhibitors, the INK4 CKIs, is a major contributor to so many human cancers.
Absolutely. Now, the key molecular target of that CDK4000 cyclin D activity is the famous RB tumor suppressor protein, the product of the retinoblastoma gene.
Okay, RB. It acts as the ultimate molecular switch.
It does. In the G0 state or in early G1, RB is largely unphosphorylated. In this state, it binds very tightly to a crucial group of proteins, the E2F transcription factors.
And when RB is bound to E2F, it's a repressor. It's physically suppressing the transcription of all the genes needed for S phase, most importantly, the gene for cyclin E.
So RB is the molecular brake holding the cell back, but when CDK4006 cyclin D activity spikes,
It phosphorylates RB.
it initiates this massive cascade-like phosphorylation of RB. This causes a dramatic conformational change in the RB protein, forcing it to instantly dissociate from E2F.
And the freed E2F flips its function. Instead of being repressed, it becomes a powerful transcriptional activator.
Exactly. It turns on transcription for all the necessary genes for DNA synthesis. Cyclin E, enzymes for making nucleotides, everything. A single controlled phosphorylation event converts E2F from a repressed break into the primary throttle for proliferation. It's an incredibly elegant switch.
And that's why a functional RB is a classic tumor suppressor. If the gene is mutated and RB can't bind E2F, the break is permanently disengaged. E2F is always free to drive unregulated proliferation.
Precisely.
Okay, so the next step is activating CDK2 cyclin E. That's the final key to getting into S phase. We know cyclin E synthesis is activated by that newly freed E2F, but CDK2 activity is still regulated by that CKI we mentioned earlier, P27.
Yes. In early G1, P27 is strongly inhibiting any CDK2 cyclin E complexes that form. But as the cell gets stronger growth signals, two things happen. First, general growth factor signaling reduces the synthesis of P27. And second, and this is critical, once CDK2 gains even a little bit of activity, it engages in positive autoregulation.
What does that mean? Does the kinase start destroying its own inhibitor?
That's exactly what it means. CDK2 phosphorylates P27, and that phosphorylation targets P27 itself for ubiquitination and rapid degradation. This creates a positive feedback loop. CDK2 activity rises a little, which leads to the destruction of P27, which dramatically accelerates CDK2 activity even more, ensuring a rapid, committed push into S phase.
Oh, now we're in S phase. The big challenge is structural. Making sure that DNA replication happens once and only once per cell cycle. You cannot allow reinitiation during the same cycle.
This is perhaps one of the most vital control mechanisms. It prevents disastrous genomic instability. The mechanism centers on licensing the replication origins. Replication begins at specific origins, but only if something called the pre-replication complex, or pre-RC, has formed there.
And what's in that pre-replication complex?
It involves the origin recognition complex, ORC proteins, and the crucial MCM helicase proteins. They are the engine that unwinds the DNA. But here's the key. This pre-RC can only assemble during the low CDK environment of G1. The origins get licensed in G1 when CDK activity is low.
But even though the complex is assembled in G1, it just sits there, inactive, until S phase begins.
That's the core of the regulation. It's the high activity of CDK to cyclin E, along with another specialized kinase called DDK, that actually initiates replication. They phosphorylate activating proteins in the MCM proteins directly, which activates the helicase. The replication fork starts moving, and the MCM proteins are displaced from the origin.
And that high CDK activity is what prevents relicensing.
Exactly. The trick is brilliant but simple. The high activity of all the CDKs during S, G2, and M phases actively prevents the MCM proteins from reassociating with any origin. Since origins can only be licensed when CDK activity is low, which only happens in G1, the cell guarantees that once an origin fires, it stays silent until the next cycle begins after division.
We briefly touched on the DNA damage checkpoints, but let's take that deep dive now into the molecular cascade that triggers cell cycle arrest when damage is detected.
This is where the cell's guardians, the central signaling kinases, step in. We have ATR and ATM. They're activated by different kinds of damage. ATR responds to single-stranded or unreplicated DNA, often found at stalled replication forks.
Okay. And ATM.
ATM is activated specifically by double-strand breaks, which are far more lethal, and the clinical relevance here is huge. ATM mutations are linked to the inherited disorder ataxia-telangiectasia, which is characterized by extreme radiation sensitivity. It really underscores its role in DNA repair.
So once ATR or ATM are activated by damage, they kick off a phosphorylation cascade.
A dramatic one. They activate the downstream checkpoint kinases, CHK1 and CHK2.
And the ultimate goal of CHK1 and CHK2 is just to stop the cell cycle. How do they do it?
The mechanism of arrest is incredibly precise. CHK1 and CHK2 strongly inhibit or degrade the CDC25 phosphatases, the very same proteins we identified earlier as the primary activators of CDK1 and CDK2.
So wait, DNA damage signaling shuts down the CDK activator?
By inhibiting CDC25, you keep those inhibitory phosphates that we want applied locked onto CDK1 and CDK2. This globally halts CDK activity, resulting in cell cycle arrest in G1, S, or G2, which provides the essential time needed for DNA repair before proceeding.
And if the damage is too great to repair, the cell often have to make a sacrifice. This decision frequently relies on the most famous tumor suppressor in all of human biology, P53.
The role of p53 is particularly crucial for maintaining that G1 arrest. Normally, p53 is made and then rapidly degraded, so its concentration in the cell is kept low. But when ATM or THK2 are activated by damage, they phosphorylate p53.
And that phosphorylation stabilizes it.
Yes. Stabilized p53 levels rise rapidly. And because p53 is a transcription factor, this increase has a direct and potent consequence. It activates the transcription of the CDK inhibitor p21.
Ah, the CKI we talked about earlier.
p21 then binds to and inhibits the CDK2 cyclin E and A complexes, reinforcing that G1 arrest. And if the damage is just overwhelming, prolonged p53 activation can trigger apoptosis or programmed cell death.
It's the ultimate quality control. It sacrifices the single cell for the good of the whole organism. And the importance here, it can't be overstated. The p53 gene is mutated in over half of all human cancers. Losing p53 function means the cell loses its ability to arrest after DNA damage, which leads to massive genomic instability and eventually tumor development.
So once the cell successfully passes that G2 checkpoint, confirming its DNA is fully replicated and undamaged, the now fully active CDK1 cyclin B complex takes the stage as the master regulator, initiating the violent cellular reorganization of M phase.
CDK1 is the director, but it immediately activates a whole ensemble of key players. It kicks off an integrated positive feedback loop with other mitotic protein kinases.
Right. CDK1, Aurora kinase, A and B, and Polo-like kinase are all activated in sequence, and then they reinforce each other. CDK1 activates Aurora, which activates Polo-like kinase, which then feeds back to fully activate CDC25, ensuring maximum CDK1 activity. These kinases then phosphorylate hundreds of targets at once to reorganize the entire cell.
Their targets are the structural proteins responsible for chromosome condensation, the breakdown of the nuclear envelope, fragmentation of the Golgi, and the reorganization of the microtubule cytoskeleton to form the spindle. Let's start with that spectacular process in prophase. Chromosome condensation.
Condensation is absolutely mandatory. The interphase chromatin condenses nearly 1,000-fold from a loose mess into these highly compact rods. This prevents the long DNA molecules from breaking or tangling as they get moved around. The force driving this is provided by protein complexes called condensins.
And condensins form huge DNA loops to compact the structure. But before that, the sister DNA strands are already linked by another complex, right?
They are. They're held together by ring-sheet proteins called cohesins, which bound to the DNA back in S phase and keep the duplicated chromatids attached along their entire length. As the cell enters M phase, CDK1 and Aurora B activate the condensins. The condensins then progressively replace the cohesins along most of the chromosome, leaving the sister chromatids strongly linked only at the centromere region. That's the final critical tether.
And at the same time, CDK1 cyclin B is tackling the nucleus itself, initiating the rapid and complete nuclear envelope breakdown.
This is a major act of structural demolition. CDK1 cyclin B phosphorylation targets the lamins, the fibrous filaments that form the nuclear lamina meshwork right under the inner membrane. This phosphorylation causes the lamin filaments to instantly depolymerize, dissolving that protective cage around the DNA.
And what about the membrane components themselves?
The nuclear pore complexes also disassemble, and the proteins of the inner nuclear membrane are absorbed directly into the endoplasmic reticulum, which stays intact. Separately, CDK1 and Polo-like kinases phosphorylate Golgi matrix proteins, causing the whole Golgi to fragment into vesicles, ensuring it can be evenly distributed later.
So while the envelope is dissolving, the cell is building the machinery for separation, the mitotic spindle.
The duplicated centrosomes, the microtubule organizing centers, they separate and move to opposite poles of the cell. This is driven by Aurora A and Polo-like kinases. They recruit massive amounts of proteins, including gamma-tubulin, to nucleate new microtubules. And crucially, CDK1 causes a dramatic increase in microtubule dynamic instability.
They grow and shrink much faster.
5 to 10 times faster than an interphase. This results in many short, highly dynamic microtubules radiating out from the centrosomes searching for chromosomes.
And the moment the nuclear envelope is totally gone marks the transition to metaphase. This allows those hyperdynamic microtubules to seek out and attach to the chromosome kinetochores.
The attachment is precise. The kinetochores of the two sister chromatids have to face opposite poles, ensuring they only attach to microtubules from opposite centrosomes. Once attached, the chromosomes start that signature shuffling motion, which is a result of mechanical opposition.
What are those opposing forces?
You have -N directed motor proteins, like dynin, pulling the chromosomes toward the centrosome pole. This is countered by +N directed motors and the growth of the microtubule itself, which push the chromosome away. The interplay of these forces eventually settles the chromosomes at the spindle equator.
This alignment results in metaphase, where all the chromosomes line up on the metaphase plate right in the center of the spindle. And here we define three types of spindle microtubules.
First, the kinetochore microtubules, which physically attach to the chromosomes. Second, the interpolar microtubules, which overlap with each other in the center and push the poles apart. And third, the astral microtubules, which radiate outward and anchor the whole spindle to the cell cortex.
The cell now stays suspended in metaphase, sometimes for a long time, just waiting for the final trials for separation. And this pause is completely controlled by the spindle assembly checkpoint, the SAC.
The SAC is the final quality control check. Any unattached kinetochore acts as a powerful sensor. It immediately generates an inhibitory protein complex called the mitotic checkpoint complex, or MCC.
And what does the MCC inhibit?
It inhibits the APCC ubiquitin ligase. The APCC cannot be activated until that signal from the MCC is silenced, which only happens when every single chromosome has achieved bipolar attachment. Once alignment is complete, the MCC signal stops, and the APCC is activated by its partner protein, CDC20.
So the initiation of anaphase is triggered entirely by APCC activity. The complex now has two absolutely critical targets that it tags for destruction.
Target number 1. The APCC ubiquitinates and degrades cyclin B. This causes CDK1 inactivation, which is necessary for the cell to exit mitosis later. Target number 2. The APCC ubiquitinates securin.
Securin, the inhibitor of the execution of protein.
Exactly. Securin is the inhibitory subunit of a protease called supprase. When securin is destroyed, supprase is instantly activated. And the newly active supprase then cleaves the remaining cohesin rings that are holding the sister chromatids together at the centromere.
That cleavage is the final molecular break. Once the cohesin link is severed, the sister chromatids are instantly free to segregate to opposite poles. That movement is anaphase. The cell is now committed to division.
Following anaphase, the complete inactivation of CDK1 allows the cell to rapidly exit mitosis. Structures revert. The nuclear envelope reforms around the two separate sets of chromosomes. Chromatin decondenses, and the microtubule cytoskeleton goes back to its interphase state.
And the final step is cytokinesis, cell division itself, which usually begins during late anaphase and physically separates the two daughter cells.
In animal cells and yeast, this process relies on the actin cytoskeleton. A contractile ring of actin and myosin cell filaments assembles right under the plasma membrane, exactly where the metaphase plate was. Contraction of this ring pinches the cell membrane inward like a drawstring and ultimately divides the cytoplasm in half.
But higher plant cells have that rigid external cell wall. They can't just pinch off like animal cells do.
No, their mechanism is totally different. They divide by building a brand new cell wall and plasma membrane from the inside out. Golgi vesicles carrying cell wall precursors are transported to the former metaphase plate. These vesicles fuse together, forming a large membrane-enclosed disc called the cell plate.
So the cell plate is basically a rapidly growing internal wall.
Exactly. It expands outward, perpendicular to the spindle, until it fuses with the existing parental plasma membrane and cell wall, successfully dividing the cell in two. And any incomplete fusion during this process leaves behind small channels, the plasmadomata, which maintain cytoplasmic connections between the new daughter cells.
We have journeyed all the way from an external growth factor signal to the physical separation of DNA, governed by layers and layers of molecular precision.
To briefly recap the absolute key takeaways, the cycle relies on the four coordinated phases, G1S, G2M. The G1 restriction point is the main decision threshold, dictated by external growth factors that drive cyclin D synthesis.
And the system's engine is the CDK cyclin system. CDK activity is balanced by the opposing forces of WEE1 kinase, which is inhibitory, and CDC25 phosphatase, which is activating.
External signals integrate by activating CDK 4006 cyclin D, which phosphorylates the RB tumor suppressor. That molecular switch releases the E2F transcription factor, which then activates all the S-phase genes.
And genomic integrity is maintained by those essential checkpoints, the ATM-ATR-mediated DNA damage pathways that halt the cycle by inhibiting CDC25 and the spindle assembly checkpoint that monitors alignment.
And the climax of the whole cycle involves the APCC, which simultaneously degrades cyclin B to allow for mitotic exit, and securin to activate supprase, which cleaves cohesin and finally initiates anaphase.
So what does this all mean? It means that every major transition in a cell's life division, growth, or differentiation is dictated by these tightly regulated, conserved molecular interactions. The cell cycle isn't a slow cascade of events. It's a series of sharp, irreversible molecular flips driven by the precise timing of phosphorylation and ubiquitination.
And here is a final provocative thought for you to explore, based on the balance of power we just discussed. We emphasize that Shijing-1 and Shicheng-Q2 cause cell cycle arrest by inhibiting the CDK activator, CDC25. We also established that the Wee1 protein kinase is the enzyme that applies that inhibitory brake to CDK1 and CDK2. So, if DNA were damaged and a cell was desperately relying on its checkpoints to arrest and repair, what might be the consequence for that cell if the Wee1 protein kinase were artificially inhibited or removed entirely? Consider that delicate balance required to maintain a G2 block before division.
Fascinating. Thinking about that balance really underscores the vulnerability of the system. We hope this deep dive into the eukaryotic cell cycle gave you the accelerated, comprehensive understanding you were looking for.
Thank you for joining us. We'll see you for the next deep dive.